Okay, so in this discussion, we will going to talk about the microbial growth. Okay, now how microbial growth is executed, and thereafter how microbes are uh, visible by means of their growth in different physical factors that we need to discuss about. Okay, but before that, we will need to talk about the remaining portion of the previous class, that is chemical sterilization, and very importantly, another thing, that is the different culture medium. Okay, in different culture mediums, artificially we can grow the bacteria in the environment, in different circumstances, in the soil and in other ingredients the bacteria may grow, but artificially in the laboratory we have to form few cultures, and by means of this kind of culture medium we can cultivate the bacteria into the laboratory. Okay, so before we proceed to the exact discussion of the bacterial growth and their physical and nutritional requirement, we need to talk about the remaining portion of the sterilization and the culture media. Okay, basically, the chemicals are also used for the sterilization. Although heating provides the most reliable way to kill the objects of all transmissible agents, it is not always appropriate because the heat may damage the heat sensitive materials such as the biological materials and many plastic components, right? So liquid sterilants and high disinfectants typically include oxidizing agents such as the hydrogen peroxide and paracetic acid and aldehydes such as glutaraldehyde. Okay, even the alcohol itself. Okay, by means of which we can sterilize different materials, and that is safer in compared to that of the heat sterilization, because heat sensitive materials cannot be sterilized by that particular process, but we can apply this kind of thing, chemical sterilants, to make the object sterile. Okay, now what is the meaning of disinfection? Okay, basically there is a difference between sterilization and disinfection. Disinfection is the process of inactivating microorganisms by direct exposure to chemical or physical agents. Now, what are the properties of the ideal disinfectant? When you can say that yes, it is a disinfectant. Okay. Ideally, the disinfectant should have a wide spectrum of the antimicrobial activity. It must be effective against a wide variety of infectious agents like the gram-positive and gram-negative bacteria, acid-fast bacteria, bacterial endospores, okay, fungi, viruses, etc. It should act in the presence of the organic matter. It should not be toxic to human, okay, or that should not be corrosive. Okay, in practice this balance between effectiveness and low toxicity for animal is hard to achieve. Okay, some kind of disinfectant is effective as well as it is, low toxic. It is very hard to find out. Okay, but that should be a ideal characteristics for a disinfectant. Some chemicals are used despite their low effectiveness because they are relatively non-toxic. It should be stable upon storage and should not undergo any chemical changes. It should be odorless or smellless or with a pleasant odor that should be present. It should be soluble in water and lipids for penetration into the microorganisms. It should be effective in acidic as well as in alkaline media. It should have a speedy action by means of its destructive activity over the microorganisms. If possible, it should be relatively inexpensive. Okay. Now, what is the difference between the sterilization and disinfection? By definition, if we go first, so sterilization means freeing an article, surface, or medium from all living organisms, including all the microorganisms like viruses, bacteria, and all these things. On the other hand, disinfection is a process that reduces the number of contaminating microorganisms that can't, you know, kill all the microorganisms present over the surface. Okay, and if we, by means of the disinfection, if we can decrease the amount of the microorganism over a surface, so that will decrease the chance of the uh, you know tendency of infection, so liable to cause infection to a level which is deemed to no longer harmful to health. Okay, spores are not killed by means of the disinfection, but that is possible in case of the sterilization. You can also destroy the endospores. Uses objects or instrument coming in direct contact with a break in skin or mucous membrane or entering a sterile body area. Disinfection can be used upon the objects or instrument coming in direct contact with mucous membrane, but tissues is intact or via intact skin. Okay, so over the intact skin you can apply the disinfection, but over the ruptured skin, over the injured skin, you have to use a sterilization process. Okay, examples: surgical instruments, needle, syringe, okay, and all these other laboratory gadgets you may sterilize. Endotracheal tubes, okay, aspirators or the respirators, gastroscopes, okay, that means the endoscope tube and endoscopy tube and all these things. Bedpans, urinals that we can disinfectant. Action of the disinfectants: they produce damage to the cell wall and alter permeability of the cell membrane, resulting in exposure, damage, or loss of the cellular contents. They alter protein and form protein salts or cause coagulation of the proteins which are present within the microorganisms. They inhibit enzyme action and inhibit nucleic acid synthesis or alter nucleic acid molecules. They causes oxidation and hydrolysis of them also. Now, what are the different category of the disinfectants? Now, here is a list of the disinfectants. Okay, includes the phenolic compounds, halogens, alcohol, aldehyde gases, oxidizing agent, dyes, heavy metals, acid, and alkalis. You just need to pause the video and just write down all these examples in your copy. Okay, and that is just for short questions. Might be they can ask you to give few examples of the disinfectants, or they may ask that name the gases or the dyes or the heavy metals which are regarded to be as a disinfectant. So just pause the video and write down these examples in your copy. Okay, nothing else.